everyone, and welcome to today's webinar on STEC confirmation using ddPCR. We're excited to have you here with us. My name is Jessica, and I'm the Director of Marketing at AMTech. I'll be moderating today's webinar. First off, we encourage everyone to participate by asking questions throughout the presentation. You can submit questions by clicking on the Q&A at the bottom of your screen, and we'll stay on after the presentation to go over any questions that came through. To start off, for those of you who don't know, AMTech is an accredited laboratory in the San Francisco Bay Area, specializing in food safety testing and special research projects such as shelf life, challenge studies, validation, spoilage investigation studies. And AMTech also just opened 2 new laboratories in Modesto and Salinas, California to better serve our clients. So if you know anyone in the Modesto or Salinas area, please don't hesitate to reach out. Today's webinar will be led by Dr. Feifei Han, Director of Food Testing at AMTech. Dr. Han boasts extensive experience working with a wide range of food pathogens, including Salmonella, pathogenic E. coli, Campylobacter, Vibrio, and Listeria mono. Her research and knowledge have been widely recognized in the industry, evidenced by her numerous peer-reviewed articles in the esteemed international journals and presentations at national conferences. During the Q&A, we will also be joined by Dr. Shiga, who is the Global Product Manager of ddPCR Food Applications at Bio-Rad. And with that, let's get started. Feifei?
Thank you, Jessica. Good morning, everyone. Thank you for joining us today. In the next 30 minutes or so, we will explore the current methods for detecting Shiga toxin-producing E. coli, or STEC, and introduce a game-changing technology called droplet digital polymerase chain reaction, ddPCR for short. We will discuss the workflow of ddPCR for STEC, confirmation, result interpretation, and present case studies to show its applications. Let's begin by refreshing our definition of STEC. STEC stands for Shiga toxin-producing Escherichia coli. It was estimated that STEC was responsible for 2.8 million acute illness annually, including 389 cases of hemolytic uremic syndrome, HUS, 270 cases of end-stage renal disease, and 230 deaths. STEC infections primarily occur through the consumption of contaminated food, including undercooked ground beef, chicken, raw milk, cold sandwiches, water, unpasteurized apple juice, sprout, and vegetables. Shiga toxin STX is considered to be the primary virulence factor of STEC and has 2 genes associated, XTX1 and XTX2. However, Shiga toxin alone is generally insufficient to cause severe illness in individuals. Multiplex genes that enable STEC strains to attach, colonize, and produce toxin proteins have been identified. In the subset of STEC strains, entrainment plays a critical role in intestinal colonization, which is encoded by YE gene. We will discuss about the XTX and YE gene in the next slides. The top 7 serotypes of STEC include 0157, 026, 045, 0103, 0111, 0121, and 0145. As we mentioned earlier, the presence of both XTX and YE gene is characteristic of STEC strains. These virulence factors contribute to the pathogenicity of these bacteria and their ability to cause severe gastrointestinal infections in humans. To detect STEC using real-time PCR, both XTX and YE genes are targeted as specific markers. In the testing process, if one of the targets either XTX or EAE is negative, the overall result is considered negative. Both the XTX and the EAE genes needs to be positive for a presumptive positive STEC. Current method for detection, isolation, and identification of top 7 STEC follow the United States Department of Agriculture, USDA, Food Safety and Inspection Service, FSIS, Microbiology Laboratory Guidebook, method number 5, MLG5. This method describes the lab procedure for analysis of raw and ready-to-eat meat products and lab environmental sponge for the 7 target STEC cell groups. This flow chart shows the procedure of USDA MLG method to detect and confirm STEC. In STEC detection process, the food matrix is initially subject to an enrichment step. Then the enrichment sample undergoes a various gene screening, targeting XTX and EAE using the VARX real-time PCR. Any sample that yield a presumptive positive result for STEC in the initial screening then proceed to the next stage, known as the Big 7 O group screen using the serial real-time PCR. If the O group screen produces a negative result, the final result will indicate a negative finding. However, if the O group screen yields a positive result, further culture confirmation is required in order to get isolate. Then the representative colony is selected for another round of various gene and O group testing. If both the various gene and O group results are positive in this confirmation step, the sample is reported as confirmed positive for STEC. One of the main challenges in the whole process is time-consuming. These methods require isolating and growing bacterial colonies from the samples, followed by additional testing for virulence genes and all antigen groups. This lengthy process may take multiple days, which present significant challenges for both the industry and the regulatory bodies when making promote decisions and implementing necessary control measures. Because of the lengthy process involved, this method is also labor-intensive. Also, multiple colonies normally needed to be picked and confirmed. The real issue, however, is the low confirmation rate for non-0157 STEC presumptive positive samples. Historically, 0157H7 has gained significant attention due to association with outbreaks and severe illness. However, the identification and the confirmation of non-0157 STEC strains have posed more challenge in recent years. The process of confirming non-0157 STEC strains often face lower conversion rate from presumptive positive to a confirmed positive result. A survey conducted in 2013 on STEC in Australia cattle faces highlight this issue. Out of the 1,500 samples tested, 44.5% were identified as presumptive positive for non-0157 STEC. However, only 1.3% of these presumptive positive samples were confirmed through culture. Regardless whether the pathogen is confirmed or not, a product may pass its prime shelf lifetime while it is pending for confirmation due to the lengthy confirmation process. You may wonder why it is so difficult to confirm STEC. The difficulty in confirming STEC comes from the concept of co-localization. STECs and EAE. Co-localization refers to the simultaneous presence of both STECs and EAE within the same cell. As we mentioned earlier, the individual presence of STECs or EAE gene is not the primary target. Instead, the target of interest is the combined presence of STECs and EAE gene within a single cell. In another word, rather than focus on the presence of STECs or EAE genes individually, the main objective is to detect the simultaneous presence of both genes within a single cell. This brings a game-changing technology called droplet digital PCR, also refers as ddPCR. ddPCR utilized phase separation of water in oil in motion to partition a sample into thousands of small volume reaction chambers. The massive sample partitioning is a key aspect of ddPCR technique. The traditional PCR, as shown in the left picture, the bulk sample provides only a single measurement. However, with ddPCR, as demonstrated in the right picture, the sample is partitioned into around 20,000 nanoliter size droplet. This partitioning enables the measurement of thousands of independent applications events within a single sample. So the real-time PCR could not differentiate between a single positive and double positive, while ddPCR take advantage of bacterial encapsulation, which allows for detection of co-localization of multiple targets, as we mentioned before, STECs and EAE gene within the same droplet. In the next slides, we will present graphs comparing the difference. The graph on these slides present comparison between real-time PCR and ddPCR for detecting E. coli 045 H18, which is STX positive, EAE negative, and E. coli 045 H2, which is STX negative, EAE positive. In real-time PCR, when these mixed strains are present, the assay will detect both genes, resulting in a positive result. However, it could not differentiate that two genes are from different cells. On the other hand, ddPCR can identify individually for the presence of STX and EAE genes. As we see from the graph, EAE is clustered with green droplet and STX is clustered with blue droplet. The orange color indicates linkage analysis between these two. The software will calculate the linkage of STX and the EAE and determine if the two targets are linked together. In this case, the STX and EAE are not co-localized within the same cell. As the result shows here, unlinked target result is negative. This example focuses on the E. coli 157 H7 strain, which has both the STX and EAE genes. With real-time PCR, both genes can be detected simultaneously, resulting in a positive outcome. ddPCR can identify and confirm the presence of the linked target. In other words, STX and EAE genes co-localized within the same cell. This ability allows ddPCR to provide a more comprehensive assessment and accurately classify the samples as positive for the linked STX and the EAE target. In the ddPCR workflow, once the samples are prepared and enriched, they will be processed through the following steps. First, the samples are introduced into a droplet generator, where they are partitioned into thousands of individual droplets. These steps takes only a few minutes. Then the droplets containing the sample are subject to the thermal cycling. This step amplifies the target DNA sequence within the droplets, generating numerous copies for next step. In the end, after the thermal cycling is completed, the droplets are transferred into a droplet reader. Based on the analysis of the fluorescence signals, the ddPCR system generates results that allow for the determination of the linkage between the target genes, in other words, the co-localization of STECs and the EE genes within the individual cells. Let's look at some case study. In this case study conducted by Bio-Rad, there was a comparison between ddCheck STEC and the USDA MLG for the confirmation of E. coli 0157H7 in 100 beef samples. These beef samples were obtained from a major producer in the US and has initially tested presumptive positive for E. coli 0157H7 using real-time PCR as the primary screening method. The result of comparison were as follows. ddCheck STEC confirmed 97 out of 100 samples as positive for E. coli 0157H7. USDA MLG confirmed 95 out of 100 samples as positive for E. coli 0157H7. This comparison demonstrates a remarkable agreement between ddCheck STEC and the gold standard USDA MLG with a 98% overall agreement in confirming the presence of E. coli 0157H7. The finding is significant and underscores the reliability and accuracy of ddPCR in confirming E. coli 0157H7 in beef samples. In another compelling case study conducted by USDA Agricultural Research Service, ARS, 100 beef samples consisting of beef trim and microtally were examined. These samples were initially tested as presumptive positive for STEC. The ddPCR technology was able to identify the presence of the linked target genes within the samples, which were also culture confirmed using the USDA MLG method. This case study highlights the power and efficiency of ddPCR in confirming the presence of specific linked target genes in STEC strains. The ability of ddPCR to accurately identify the co-localization of STECs and EAE genes provide valuable insight into the pathogenicity and virulence potential of these strains. In summary, today's webinar shed light on the challenge posed by traditional methods such as real-time PCR and culture for confirming STEC. These methods are time-consuming and may not provide insight into the co-localization of important virus genes. However, a game-changing technology, ddPCR, was introduced as a solution. ddPCR enabled the confirmation of both STECs and the EAE genes within the same bacterial cell. The implementation of ddPCR holds great promise for both the food industry and the regulatory agency by providing more accurate and efficient STEC confirmation. ddPCR can significantly benefit the industry by streamlining testing protocol and improving overall food safety. Regulatory agencies can also leverage ddPCR to enhance their monitoring and surveillance efforts, ensuring better control of STEC-related risk. Thank you for your attention. If you have any questions, I will do my best to provide answers.